Thank you so much. Thanks for inviting me to speak here today about inclusion of pregnant and lactating people in clinical trials. This is a terrific session, and I wanted to, in particular, thank Lori, who spoke today. And so Lori would be the type of patient that I would see in my clinical practice as an obstetric medicine physician at the McGill University Health Center, and is exactly why sort of I'm here and talking to you today. So just a few words on disclosures. So I have received operating funds from CIHR and the Heart and Stroke Foundation of Canada for my research, as well as support from the Fondation de Recherche Québec en Santé in the form of a bursary, and of some honoraria, as you could see. So I will stay on time, and we have a few learning objectives, but they, of course, will be brief. So we'll briefly just review some of the ethical issues of inclusion and exclusion of pregnant and lactating people in clinical trials and a bit of the historical perspective. I'll be also briefly discussing some examples for non-communicable diseases as well as communicable diseases that highlight these inequities, understand the barriers to inclusion and what we can do about it, and I'll briefly introduce the PREG trial initiative. So how did we get here? So a little bit about the historical perspective, and some of this was touched on already. And how many of you know about thalidomide? Right. Okay, so it's like a pretty famous story in medicine. So, you know, the thalidomide crisis of the 1950s, thalidomide was a drug used to treat nausea and vomiting of pregnancy. And in the 1960s, it was uncovered that as many as 10,000 infants were born with pretty severe birth defects affecting their limbs. So obviously this was a tragedy, and it was removed from the market, although it still remains as a drug for multiple myeloma. And there's some consequences of this crisis. Some of them are actually positive. So one is that the development of more robust toxicity testing really occurred after this, the thalidomide crisis, both in animals and humans. Also, pregnant patients were labeled as a vulnerable group. And we'll talk about how that's a double-edged sword. And I think, you know, that's sort of why we're here today. And finally, there was a fear of not only prescribing, but testing novel drugs in pregnancy. So in the 1970s, the FDA introduced the classification for drugs in pregnancy. And again, many of you probably know these: category A, B, C, D, and X, A being safe and X being teratogenic. And so while we find this, you know, in obstetric medicine, we now have sort of moved away from FDA, as it is rigid and doesn't incorporate up-to-date guidance, but it did, at the time, it was a way of offering guidance for health professionals and introduced this concept of balancing risks and benefits. And finally, now we're in an era of advocacy in so many different spheres, but in particular in this area. And so the pendulum has swung towards advocacy for inclusion, which is so important. And I should really include here Bridge, which is a fantastic initiative that was spoken about before the break. And other organizations such as the FDA, the National Academy of Sciences, which have recently released very robust and detailed guidelines on how to include safely pregnant and lactating women in clinical trials. There was a Tri-Council policy statement in Canada as well about 5 to 10 years ago. And Dr. Malhame, who will be speaking after me, will talk a lot about how we can advocate as individual scientists and as clinicians. So all of this is really great. We're moving in this direction now, and we need to actually move beyond advocacy towards action. So when we talk about prescribing in pregnancy, when we talk about inclusion of pregnant and lactating individuals in clinical trials, we really need to understand the maternal context. So some maternal factors that really are important are that individuals now that are pregnant, they're entering pregnancy with obesity, diabetes, hypertension, so metabolic condition, psychiatric or mental health issues, autoimmune conditions such as the patient speaker today. And these are really, really common in individuals entering pregnancy, probably more so than in the last 30 years. And of course, the management includes pharmacology. There are also some infectious diseases that disproportionately affect pregnant individuals and their offspring, such as influenza, COVID, which we'll hear a lot about, RSV, Zika. And these, of course, not only require management when the conditions arise, but prevention through vaccination. And the reality is, and I know this was mentioned already, that 70% of people enter pregnancy taking at least one prescription medication, yet this is without sufficient data. So what are some of the consequences? The consequences are we're using medication without high quality evidence. We simply are. we are either undertreating or mistreating patients during pregnancy and lactation. So withholding medication that we don't have enough data about that would actually benefit the patient, or just using another agent that really wouldn't be as effective. So obviously there's a risk of disease flare, which is associated with poor outcomes in pregnancy, and even a risk of death. So the confidential inquiry into maternal mortality in the UK has uncovered withholding of necessary medication for chronic disease being directly attributable to some maternal mortalities. And pregnancy and lactation are transient states in a woman's life. But what I would like to underscore is that there's no access to evidence-based care in some cases during all of the reproductive years. So if someone sort of seeks preconception care, they might be told not to use medication X. Sometimes it's appropriate, sometimes not. So while they're planning pregnancy, then while they're pregnant, then in between pregnancies, then while they're lactating. So many years go by and they're not receiving evidence-based care. There's also an impact on breastfeeding, and this is spoken about a lot less. Even though most drugs enter breast milk, very small quantities enter breast milk, and most are by and large safe. That being said, there's a lack of pharmacokinetic studies or trials including lactating people, which results often in premature breastfeeding discontinuation. We don't know enough about this molecule, so individuals are asked to stop breastfeeding before they really have reached maximal benefit, or inappropriate cessation of beneficial medication during lactation. So both of these are obviously suboptimal. So an example, a non-communicable disease or a chronic disease that many pregnant individuals have when they enter pregnancy would be diabetes. And I just want to give a little bit of context here on some medication and what the trajectory has been and how this has been underscored some inequity. So metformin, 20 years ago, observational data which showed, through meta-analysis, showed by and large safety of this molecule without any signals for teratogenicity or fetal toxicity. That was 2003. There were then several clinical trials on use of metformin for gestational diabetes and for type 2 diabetes over the last 20 years, as well as post-marketing surveillance of the children born to these mothers. While there was possibly a signal for smaller babies, it was otherwise found to be extremely safe, including in follow-up studies. It is still not in guidelines as an option to continue throughout pregnancy. Metformin, first-line therapy for type 2 diabetes. We've had to spend 20 years and all of the funding and resources on studies of metformin before we could even say, okay, you can use this in pregnancy. And metformin actually minimizes use of insulin, minimizes weight gain, has a lot of benefits to it. So, metformin is one kind of example. Novel diabetes drugs, GLP-1 agonists, SGLT2 inhibitors, those of you who are in the adult medicine space know about these drugs. They've changed the landscape of diabetes care. They result in significant weight loss, cardiovascular and renal benefits, mortality benefits, excellent glycemic control. But because trials have not included these individuals during pregnancy or lactation and minimally include reproductive age females, not enough studies looking at the quantity in breast milk, we really can't advise patients on use of this in their reproductive lifetime, and that it means that they're being undertreated for type 2 diabetes during their reproductive period. So the ethical issue is that effective treatments are withheld from pregnant and reproductive age females, and this is not equitable. A communicable disease example, so COVID-19, when the pandemic first started, as we were gathering data, at first it didn't seem as though pregnant people were at particular increased risk, but it didn't take long. Basically 6 months in, it became very clear that pregnant patients were found to be at increased risk of severe COVID-19 disease, and 3 times increased risk of an ICU stay, preterm birth, and severe maternal morbidity. And early on, there was a systematic review in the pandemic showing that only about half of the ongoing trials were enrolling females of reproductive age. The majority excluded pregnant people, including studies of drugs such as hydroxychloroquine, which already has a safety record in pregnancy. So lack of trials really perpetuated the risk of severe disease. And then the risk, we talked about the risk falling on the patient's shoulders. It really does fall on the clinician's shoulders as well. When faced with a pregnant patient who's sick with COVID-19 or whatever else and has to make a decision, about, using a medication to save their life that has no real evidence in pregnancy, that's shifting the risk from the pharmaceutical industry to the clinician and the patient. And then, of course, vaccine hesitancy and delays in inoculating pregnant patients can be due to the fact that in the vaccine trials, pregnant women were not initially included. So I spoke really, those two cases illustrate some barriers. The barriers are at the level of the drug company, who perceive legal risk and rightfully so realize that there will be delays in study rollout if they include pregnant patients. And a lot of that is because they're labeled as a vulnerable group. Clinicians lack knowledge and comfort enrolling pregnant patients. Researchers have identified that there are ethical complexities at their local REB boards. And I love that Dr. Mitra discussed the different forms of consent, because that's really needed. And there's a lack of kind of harmonized resources, tools, and processes that can be used in this population. There's insufficient funding, not just in the amount, but in the time. So to enroll pregnant or lactating people and ensure we have enough time to follow them in the post-marketing phase requires not just a 4-year grant, but like an 8-year grant. So you really need a lot more funding. And then at the level of the patients, there's fear, there's a misunderstanding about trials in general, the benefit of trials, and about drugs in pregnancy, and informed consent has been inadequate. So how can we address these issues? So this really terrific proceedings of a workshop done in the US, the National Academy of Sciences, led by Dr. Ruth Faden at the Johns Hopkins, really outlined very actionable and concrete recommendations for how to address some of these issues. Reducing regulatory barriers is number one, and that's not just at the level of the FDA, but that's at the local REBs. That means actually removing the label of vulnerable patient, because it's no longer serving pregnant patients. Enhanced training of researchers and staff, just like IMPaCT is doing. Funding, as I mentioned, for post-marketing surveillance. Investing in tools to assay drug levels in breast milk. Innovative study designs, so adaptive study designs. Adapting consent processes and tools, and also multinational collaboration, which is really, really critical, as obviously small sample sizes in a given trial is not going to generate the efficacy and safety evidence that we need. So an example of an innovative study design, Scott Halperin, who is a Canadian researcher and works in vaccine research, actually has written about this. And these are the phases of a clinical trial that we all know about. Phase 1, which is really the dose finding and safety phase, and then phase 2, where you're testing effectiveness and further evaluating safety. And phase 3 is usually the multi-center efficacy trials. So a safe and also resourceful proposal would be to embed phase 1 of pregnant people into phase 2 or 3 clinical trials for the general population. So that adds an additional sort of safety net, and yet does not require us to wait, all the way until post-marketing phases to know if this is safe or effective in pregnancy. So some considerations of how to appropriately include patients in RCT study design. So it doesn't mean default inclusion for all. It just means consider inclusion. And some questions to consider are, does this condition substantially affect reproductive age pregnant or lactating women? Obviously, it wouldn't make sense for hemophilia to include pregnant patients, right? So it would be cardiometabolic, reproductive, mental health issues, those that really are should be prioritized. Is standard of care inadequate? Does animal data suggest harm or teratogenicity? Obviously, we would avoid in those situations. Is the dose or frequency similar to pregnant women and non-pregnant? Will this trial alone be able to recruit pregnant or lactating women? And is there funding to extend post-marketing surveillance? So just want to end by saying changing culture is complicated. I'll briefly describe the PREG trial initiative that I'm very thankful that IMPaCT has supported, and others in this room. So, when we talk about changing culture, when we talk about moving from advocacy to action and really, really getting that knowledge out there, we have to know who we're dealing with. And there's multiple stakeholders, as was really mentioned, and the way we engage with these stakeholders should be different and really depends on their current perceptions, preferences, and strategies. So PREG trial is an initiative. It's really an implementation science project in which we hope to remove barriers to the inclusion of pregnant and lactating people in clinical trials. working in two different themes, non-communicable and communicable diseases, in which we hope to synthesize and consolidate the existing ethical frameworks and creating one that's appropriate for use in the Canadian setting. We're working with the Accelerating Clinical Trials Consortium and looking at all the IMPaCT clinical trials in Canada and doing an environmental scan of who's including pregnant and lactating people, what are some lessons that we can learn, and basically use those resources. We'll also be assessing barriers and facilitators among researchers in the IMPaCT consortium, and then finally developing tools. So really looking forward to the discussion later so that we can really decide what are the outcomes, how should we be framing informed consent, and there should really be a standardized tools, including applications to REB, Health Canada, and consent forms. So in summary, pregnant people do require pharmacotherapy. They're often prescribed medication without an adequate evidence base. This is important for non-communicable as well as communicable diseases. There are many barriers. Strategies to change culture must address all of these barriers, and there are many positive initiatives underway. And I'll just conclude with this quote. If we consider the availability of a drug proved to be safe and effective through the devices of modern clinical pharmacology and clinical trials a benefit, then it is unjust to deprive classes of persons such as children and pregnant women of this benefit. So with that, I thank you for your attention and looking forward to the discussion.
Hello, everyone. Thank you so much for the invitation and the opportunity to talk a little bit about this topic that I'm really passionate about as well. I'm here talking to you as a clinician scientist. In my day-to-day practice, I see people before, during, and after pregnancy who have chronic medical conditions or new conditions during pregnancy. So a lot of times I'll be counseling pregnant patients or people, looking to become pregnant about how to manage their underlying conditions, and that involves a lot of medications that they're currently using. So this is part of my day-to-day practice. I'm also talking to you here as a person with lived experience of pregnancy and current living experience of pregnancy. So if I do sound short of breath, it's because I am short of breath. Yes, so I will start with a land acknowledgement. We're here in Vancouver, but I acknowledge that the land where I live and conduct my work, Tiohtià:ke, is the traditional and unceded territory of the Kanien'kehá:ka, and it's a place that has long served as a site of meeting and exchange amongst nations, and that continues to carry this tradition of cultural mixity forward. And it's particularly important, I think, for our work to recognize the structural determinants of health that we are dealing with in healthcare and in healthcare research, and to continue to honor and recognize the Indigenous ways of knowing as they refer to reproductive health. So at the end of this session, you will hopefully all be able to reflect on lessons learned during the COVID-19 pandemic about inclusion of pregnant patients in clinical trials, identify perceived barriers and facilitators to the inclusion of pregnant people in clinical trials, and anticipate some of the practical aspects related to the inclusion of this population in clinical trials. So we'll start with a case study that you will all perhaps be familiar with. We're in December 2025, and the World Health Organization has declared a pandemic of influenza H7N9. And we suspect that this disease might be more severe and affect pregnant individuals differently. And as a person with an interest in clinical trials in pregnancy, you have been asked to join as a co-investigator on a multi-center trial that will be investigating the use of oseltamivir to reduce severe pulmonary disease. The trial's steering committee is reflecting on methods to support the inclusion of pregnant individuals in this trial. And so you're wondering how we should go about including pregnant participants going forward. So what we've learned, and as you've heard from the talk earlier, during the COVID-19 pandemic, despite more severe outcomes in pregnant individuals, and despite everything that we know about the absolute need and moral imperative to include pregnant and lactating people in clinical trials, most trials, at least early in the pandemic, did not include pregnant individuals and specifically excluded them. And importantly, these were trials that were sometimes looking at drugs that already had a track record of safety and efficacy in pregnancy that could have been part of consent procedures and could have been used to inform eligible and potential research participants. And that includes chloroquine and hydroxychloroquine, which has long been used for the treatment of arthritis, lopinavir and ritonavir that has been used for the treatment of HIV for many, many years, and convalescent plasma studies. And we know that these products can be administered safely in pregnancy. So we're not talking about funky phase 1 and 2 new novel trials. We're talking about bread and butter medications that are used in pregnancy. What we've also learned is that despite the fact that they were actively being excluded from clinical trials, they were also given investigational drugs outside of the purview of a clinical trial. So that includes drugs that were later on found to be associated with severe adverse events, including arrhythmias, allergic reactions, etc. So they were given these medications sometimes as an end, a third line medication, but really in the absence of any evidence for using these medications. And we really have proof that these were being given in the United States, including, in places where there's a very high litigious potential. So they're excluded from trials, yet we still gave them the medicine without all the monitoring that a trial infrastructure would provide. And that is very problematic. Eventually, though, we learned, and some trials did change their inclusion criteria, and many allowed for pregnant individuals to be eligible and to be included in clinical trials, including trials that were investigating the use of remdesivir, convalescent plasma, and anticoagulants. And I'm just showing you here a picture of our management order sets or our clinical guidelines at the hospital where I worked, where as part of routine clinical care, trial participation was offered for all patients admitted with COVID-19, including those who were pregnant. So there was an evolution throughout the pandemic of how we were approaching this topic. So what we were interested in early on in the pandemic is understanding what are some of the physician perspectives on the inclusion of pregnant people in COVID-19 clinical trials. And the reason why we surveyed physicians specifically, while there are many other stakeholder groups that should be surveyed about this, is because physicians belong to a professional group with the potential to really shape the inclusion of pregnant people in clinical trials because of so many investigator-initiated trials that are important in this area and that oftentimes will be led by clinician scientists that are physicians. Physicians' opinion as well may affect the motivation to design such research, and pregnant people may look up to their physician for guidance about participating in clinical trials. So this is really a first step. We really surveyed a small portion of the population of stakeholders, and there are limitations to that. But given that, we developed an online questionnaire with a multidisciplinary team. We assessed the perspectives of physicians that were surveyed on the appropriateness of inclusion and the urgency of including pregnant individuals in COVID-19 trials, their level of comfort with such enrollment, and some of the barriers to inclusion. The survey was pilot tested among 10 physicians from different specialties, and it was disseminated across professional societies, including the Society of Obstetricians and Gynecologists of Canada, which was facilitated at the time and still is being facilitated by Jocelyn Cook, who might be in the audience here, the Canadian Society of Internal Medicine, and the Association of Medical Microbiology and Infectious Diseases of Canada, as well as the Canadian Critical Care Society. So these were felt to be physicians who were likely to interact with patients who may be critically ill with COVID-19 or at least hospitalized with COVID-19 and to be pregnant. And so we had 168 respondents, so quite a good snapshot of our population of physicians. Mostly, we had responses from obstetricians and gynecologists and maternal fetal medicine specialists, but we also captured the opinions of people in family medicine, infectious diseases, critical care, as well as obstetric medicine and general internal medicine. People were working in academic centers in majority, but also in community hospital setting, as well as primary clinics and outpatient setting. And while some were, had a portion of their practice dedicated to research ranging from 25 to 75% of research protected time. Most of our respondents were people who in fact did not have protected time for research, so who really represent the bread and butter of clinicians who may come across inclusion criteria for a clinical trial. And what we found is that most respondents really supported the inclusion of pregnant people in clinical trials. That included that they found that it was appropriate for them to be included, especially if there was a safety track record of the medication in pregnancy. And actually, a lot considered that it was critically urgent or highly urgent to, provide this inclusion going forward, and that they were more or less comfortable with doing so. When we looked at the answers of those few who did mention not being comfortable, finding it non-urgent or inappropriate, we found several expressions of reluctance. And this is really important because, as you know, these are some of the barriers and the issues that really will need to be addressed in priority in order to be ready in 2025, 2026, when the next one happens. And so these are not surprising responses, but potential harms to the fetus, adverse effects on the mother, perception of low risk, so communication surrounding the risk of the disease in pregnant individuals is particularly important. Also for trial concerns, the lack of demonstrated efficacy, which when we have clinical equipoise, this is very common to not know whether the drug is going to be working or not. The culture of non-prescription, so we're really talking about a broad culture change, the lack of experience with the medication, and the lack of understanding of the natural history of the disease. So these were expressions of reluctance and barriers that were specifically identified were the perceived unwillingness of other team members or the perceived unwillingness of pregnant individuals to participate in clinical trials. But for those of us who have worked in partnership with pregnant individuals who do clinical research, I really want to emphasize that this is a perceived unwillingness, because a lot of time there will be an overwhelming willingness to contribute to research and to participate and to join as active members in research. So this is, again, a bit of a miscommunication between what is being perceived in the physician community that answered the survey and what is actually going on in real life. Many highlighted the burden of regulatory approvals, where even though the researchers themselves were really interested in conducting trial that included pregnant individuals, their REBs and Health Canada was actually the ones blocking the process. So one may ask, who's being protected if really, patients would like to participate and would be protected from some harm by participating, and researchers are willing to go the extra mile, and the REBs and Health Canada are protecting. We can just have an interrogation there. And again, the burden of recruitment and the lack of eligibility of participants were some of those barriers that could be addressed going forward, and liability concerns as well. Finally, the lack of infrastructure, where the individual physicians were thought to be very alone when facing the potential for recruiting pregnant participants. So our take-home messages from this survey was that, of course, there's a need for increased continued education about the potential benefits, and to really highlight the importance of self-determination for potential pregnant participants, and to facilitate this inclusion, the barriers that have been identified really need to be addressed. So at this stage, what do we need, and how can we still walk the walk to include pregnant participants in non-obstetric clinical trials? And what I mean by non-obstetric clinical trial here is a trial that is not primarily designed to answer a question about pregnancy, but a design that is currently enrolling adult participants for other reasons, and that we want to make sure that this trial does not leave pregnant individuals behind. And I'll walk you through a practical example of our SNAP trial, which is a trial looking at different interventions for the treatment of Staph aureus bacteremia. And the reason why we wanted to write this piece was because of the challenges in conducting clinical trials. And we wanted to be able to share our lessons learned by our group by adapting such a protocol in order to potentially help other teams do the same with their own trial. So the first step was really about our team, building an interdisciplinary working groups that will include persons with lived experience of pregnancy to really be able to capture what are going to be the complexities of designing the pregnancy-specific arm of the trial. And so it's important to work in an interdisciplinary fashion with people who can understand both the trial content and the diseases being studied in the trial, as well as the whole pregnancy piece, so we can all come together and strategize. We also want to be early on identifying an independent pregnancy-specific drug safety and monitoring board, so that in case there are some adverse events, we can really look at them with the lens of people who understand both worlds. For eligibility, we want to define eligibility criteria for the pregnancy arm, be it self-report or the result of a test that may support that the patient is actually pregnant, and include pregnancy-specific information about the intervention within the usual consent procedures. So for instance, when we were doing the SNAP trial, we really included data specific to the medications that were being studied as they related to pregnancy and what would need to be known at the moment of consenting from a pregnancy perspective, but it was just like an adjunct to the usual consent procedures. Then for the dosing, we heard that dosing is important in the NICU. Dosing is really important everywhere, especially when taking into consideration physiologic changes of pregnancy. You want to make sure that your intervention, if it's a pharmacological intervention, is really adapted to pregnancy specifically. And importantly, decide on some of the trial endpoints that may be specifically pertinent for pregnancy-specific research. So that includes actual pregnancy outcomes, safety outcomes that may be particularly important for pregnancy that are different from the other trial outcomes. So for instance, because of the increased prothrombotic risk in pregnancy, we are really interested in capturing the number of thrombotic complications associated with some of the medications that are being given intravenously for the SNAP trial specifically, as well as neonatal outcomes. And all this needs to be done in balancing the added burden of data collection. So we want to know more, but we also want to make sure that everything that we are going to be asking in terms of additional information will really be relevant and will inform clinical practice. So that has been our proposed approach for the trial that we adapted for the inclusion of pregnant individuals. Of course, each of these points need to be adapted for individual trials, but that gives us a general framework of how this can be done, and this has been done. We did adapt the trial to include pregnant individuals. 5 countries are now recruiting pregnant individuals, and we do have some that have consented to participate, and we have, as part of this SNAP trial now, data that is going to be specifically collected for pregnant individuals. So the take-home message is that study protocols of non-obstetric trials can be successfully adapted to accommodate pregnant participants. It can be done and it should be done. We should always have an assumption of eligibility instead of a culture of exclusion going forward. And when December 2025 arrives, we need to be extending our learnings from the pandemic to non-COVID-19 trials. We need to offer participation in non-obstetric clinical trials, and particularly in trials that are going to be addressing severe and potentially life-threatening condition. There really is a moral imperative there, and that with careful and diligent planning by a multidisciplinary team really dedicated to considering pregnancy-specific complexities, this really can be done. So I thank you very much, and I'll be happy to answer any questions.
Thank you to the organizers to provide me with the opportunity to attend this, to co-learn and to get to know the real impact of IMPaCT. So congratulations on that. I'm really, really impressed. I'm Karel Lagarde. My background is neonatology and clinical pharmacology. I live in a very small country, Belgium, where you can actually drive through, respecting the driving restrictions, in 3 hours, which is somewhat different from this country. And I actually said during coffee break that I forgot to check where Vancouver was on the map. And actually my wife explained me before I'm leaving that this is really much further down than Toronto, or so that's what happens. But I've been asked to talk to you a little bit about, let's say, real world data and PK data to inform the design. And so it has already been touched on that pregnancy and lactation are specific transient physiological settings who in themselves may affect pharmacokinetics as well as pharmacodynamics, like for instance, autoimmune diseases or diabetes. You could imagine that these thresholds are not only just simply extrapolated from non-pregnant women. I have nothing specific to disclose except the fact that off-label use will likely be discussed. And for the sake of time, I tried to restrict a little bit my message to 3 bullet points. And one is we can be very pragmatic. And that's a little bit also how I understood the previous talk. We can include pregnant women in already existing trials. But we can actually also make trials that we can include a lot of different type of drugs exposures during pregnancy or during lactation and still have one single trial agreed on by the ethics committee or the IRB board. Second, we should perhaps be more accurate and complete. There is a zillion of data out that are not as useful as we would like them to be. Statement like, we were not able to observe the drug in the human milk. Thank you, but it's not really informative if you do not add the quality of your assay. So how low could you measure that? What you have done, actually, we are not able to measure any concentration below limit of quantification of observation. That matters, and I will explain you to a certain extent why. And then the most important point was believe in science. Believe in multidisciplinary science, because together we are stronger. And we would like to show you a little bit with PBPK modeling and also even more with physiological-based modeling. We can actually create an in silico pregnant woman, throw the drug in, and predict how the drug will most likely behave. And that's an important link with the previous speaker, because if you want to do a study design, if you want to do a PK/PD study for a given drug during pregnancy, it's terribly important to a priori select the most likely best scenario. If you have a trial with 200 pregnant women and at the end of the study you see, well, this trial didn't show any difference, but actually we were given the wrong dose, it would be a pity. And we have done that. I will illustrate that. So we have learned along the way and we should implement that. First message, be pragmatic. This is something that we just started about a year ago. We call it the umbrella study. And actually any mother who is breastfeeding can be included in this trial. It doesn't matter which drug she takes. We can simply assess, do we have any information on this drug already? So we don't need novelty if we already have 30 or 40 cases described. Second, we can discuss with the parents or the subject, what do you prefer to do? Do you agree that we collect blood samples in you? Do you agree that we collect samples in your infant? Do you agree that we collect mother milk? And if so, what is reasonable? Taking also the scientific background, like for instance, elimination clearance type of ideas. And do you agree that we collect safety data in you and or your baby? And then we have something like a tick box. Because sometimes mothers agree that the blood sample is taken in their infant, or perhaps there is a clinical indication to collect so. And then you can adapt your clinical indication to your clinical needs. Quite common, they prefer not to, and that's okay. But in this way, you can be very pragmatic. And that's the very reason that we also published this protocol, because we thought, okay, this is just Belgium, 3 hours drive and you're through. But in fact, we already recruited patients in London. You take the train, in 2 hours, you're in London. A mother was taking clopidogrel, contacted us and say, well, I would like to contribute. And then we went there and collected all samples at her home. So my PhD student enjoyed 3 days of staying in London and came back with the samples. And actually, to put even more bizarre perspective, we have a patient now in Finland who has contacted a Texas group for advice on a given drug and say, well, we cannot help you. Well, they could advise, but if you would like to contribute to a trial, there's another group in Europe working on that. And from a US perspective, Belgium and Finland is close by. It's not really close by, and we have some burdens, but actually we recruited this patient and she's collecting every month. It's a very slow, very long elimination half-life compound. And she's collecting every month. And we'll go and get the samples somewhere in August. So in this way, you can actually be very flexible and ask parents also, what do you want? How far do you want to go? And what is a reasonable commitment for you? And like, for instance, the woman said, well, let's wait. I will do a first round. And if this is okay, I'm more than happy to give a second sample. And so this flexibility does provide much more comfort to both us as researchers, as well as to patients or families involved. So this works in our hands. It's a public domain, so you can simply take it and also do it in Canada or wherever in the world. The second point that I would like to stress is be accurate and be complete. And I simply would like to illustrate this based on a recent analysis, or relatively recent, on lactation and antidepressants, where a significant number of papers were excluded because it didn't provide sufficient information. And that's a pity, because I assume that the researchers who published this really wanted to contribute, and assume that the patients who contributed samples also wanted to be their contribution impactful. So how come? And I think even for, let's call it simple case reports, there's some guidance. The colleague Anderson has reported, or have provided, in my opinion, a useful suggestion, and we try to stick to that. Report as much as possible data. What do you would like to see in a case report to make it as useful as possible, not only for yourself, but perhaps also for future pooling and making more sense of it. And my third and most important point is belief in multidisciplinary science. And as I was already alluded to, pregnant-related physiology changes, kidney function changes, body size and body composition changes, and also liver activities, enzymes change. This is not a dichotomous covariate. It's in general rather trimester mediated. So you're not pregnant or not pregnant, but it's a little bit more complex. But the good news is, and it's very similar as what we're doing now in neonatal, we can use this type of data. So even basic case reports of limited number of data would inform us. And just to illustrate it, I've never been pregnant myself, but I understood that a lot of women do not tolerate caffeine during pregnancy. Well, this can be explained because if you look well, on general, liver activities will increase during pregnancy, but there are some weirdos who do not, like CYP1A2, which is exactly the one who will metabolize caffeine. So you can exactly explain this. Your clearance goes down because you're pregnant. Hormone mediated. And so intestinal functions change, liver function change, kidney function change, not just GFR, but also transporters. But we have learned how they change. And again, you can actually make an in silico, so a computer-driven system, where you can put all these covariates in. And you can actually also say, okay, this is a normal pregnant woman. Imagine that this is a woman with severe inflammation or obesity or other type of things. So you can add specific characteristics and further modulate your model to predict this more better. So there is a lot of strength in science to guide you in your clinical trials and actually to assist you in your study design. And there's a strength in it because we all know, for those who work in research, if you have a hypothesis, you can prove or reject your hypothesis. So if your hypothesis is, I have to give 1,000 milligrams instead of 800 milligrams to attain a similar exposure, you only need a few subjects to prove that you're on target. So that's the strength of this type of models. Again, we have learned in the meanwhile on the physiology and the changes throughout pregnancy, as I have also illustrated on this slide, and we can use these type of things. Does it matter? The previous speaker also alluded to infectious mediated aspects, and this is another illustration going back in 2007 on a very rare antibiotic, amoxicillin. At that time, there was no guidance how to dose a pregnant woman with amoxicillin. And actually, this paper has been done because at that time, there were some concerns on anthrax. So sometimes infectious diseases or concerns, we can also move a little bit the needle into the right direction for pregnant subjects. As you can see, this is the simulated exposure in the second, third trimester versus postpartum. I assume the majority of you know that you have to attain a given exposure to be an effective antibiotic. And actually, the normal doses are way too low for the second and third trimester of pregnancy. You can say, so what? Well, the funny thing is, for those who work in perinatology, they may be aware of the pivotal Lancet Oracle studies, who try to assess the impact of antibiotics for threatened labor and or without ruptured membranes, and whether antibiotics for 1 week were better or worse on the outcome of the pregnancy and the newborn. But post hoc, they simply used the wrong dose. It's unfortunate, but actually, post hoc, we should repeat these studies because at least what we know is that the exposure would have been way too low to have any therapeutic effect. And exactly the same is so for monoclonals, new hype in medicine, in human medicine and pharmacotherapy. We know how these monoclonals to a large extent behave. And in this way, different monoclonals, if you put them together, they always will learn you something on how does an average monoclonal will behave in a pregnant subject. And how can we translate it for even a drug that we have never used until now in a pregnant subject. So that's actually the main point that I wanted to make. The placenta is a really interesting organ. It's not a coffee filter. It's much more complex. There used to be a time that people thought nothing comes across the placenta, but who would be so stupid because the fetus would never grow if nothing goes to the placenta, right? Then we thought it's just all passive diffusion, but there are a lot of things are happening over there: metabolism, active transport, active efflux, and we start to learn also on all these different type of expressions patterns throughout pregnancy. And in this way, you can predict, like for instance, for opioids, that if you have a genetic more active transporter or less active, that the exposure to the fetus will maybe likely different, and the likelihood of abstinence will be higher or lower. So we start to know more and more on how these different type of drugs behave. And this means that we can also predict to a large extent, based on the physicochemical characteristics of this drug, how does this drug behave? How water soluble is it? Is it metabolized? Is it by a transport or whatsoever? You can predict with a reasonable likelihood how it will behave. And exactly the same, and this is something that I learned over the last years, is the beauty of the breast tissue. Because also over there, breast tissue is neither a coffee filter. You have, again, it's a little bit the way I try to teach it to my students, is say the breast is like a kidney. And then they look a little bit weird. But the point that I want to make is that at the breast, you have first passive diffusion type of patterns. And then you have a lot of modulations, just like you have in the renal tubules. Active secretion, active resorption, and other type of stuff. So you can actually already predict to a large extent how much of a given drug, even if you don't have yet data, you can predict to a large extent what will be the most likely exposure. Exposure obviously doesn't equal exposure to the infant, because if the drug is not absorbed, it will not really make it into the baby. So a drug can be in the human milk, but may not be absorbed in the baby, and so therefore the consequences could be significantly different. I already alluded to this in silico type of models, and this is how a pregnant woman looks like. It's boxes. So you have, for instance, sorry. But you have a given blood flow to the lung and a given blood flow who comes out of the lung, and you have a given amount of lung, and you could have there a lot of enzymes who will metabolize your compounds. Same for kidney, brain, or whatsoever. You have a placenta, and you have all this type of stuff. And actually, you can average the weight and the size and the volumes. This sounds perhaps weird, but this is at present already commonly used for drug registration. If we discuss drug-drug interactions or drug-food interactions, a lot of companies will submit to FDA, EMA, or Health Canada this type of information. So this is not a new invention. This already exists as a concept in current drug approval and drug development. So to bring it to a next perspective, what if you can combine in vitro data, cell models, with in vivo models, animal models, with human findings? In this way, you can really learn much faster and have the best selection of a clinical trial. This is what we have done. Unfortunately, the study never took up because EMA decided that sildenafil can no longer be given to pregnant women, irrespective of the indication, which is, don't tell it anywhere else, but it's stupid. So we try to convince them that indication also matters in life, but you can actually predict how sildenafil will behave, and this way can predict how sildenafil will make it to the fetus. The background is we want to develop a new treatment for diaphragmatic hernia. And obviously, if you give it in healthy fetuses, as well as in animal models, their lungs are poorer off. In hernia, they are better off. So that's the reason that we want to convince still EMA that they have to reconsider their position. And exactly for pregnancy, you can also develop such models for lactation. You can have in silico, you can have in vitro models, cell models for transporters. You can have animal models. What we commonly use is the pig model, but there are also other animal models. And you can actually make sense from 1 or 2 case reports of clinical observations to bring all information together and estimate most likely scenario. At present, we are in negotiation. We will not make it. Negotiation with EMA to say, well, if you would use this model, you can actually predict once a drug comes on the market, this is how it will likely behave. So you can actually, with a reasonable scientific confidence, say the likelihood that this drug will be relevantly exposed in human milk is very low anyhow, which would be very informative for clinicians and families who have to make decisions. You can also use this for recreational type of things, like cannabis. You have a mother compound, you have a baby compound, you have a milk compound, and then compartment, and you can actually predict how it will behave, how it will appear in breast milk, and how it will appear also in the baby. So along these lines, the real world data, in my opinion, matters because it's really data integration, lactation-related infant physiology and postpartum maternal physiology who will assist you to make this type of lactation models more relevant. We have to be aware that the baby who's having breastfeeding is not the same average baby as a baby who receives formula. And so one of the things that we are currently working on is develop a model that is actually a breastfed baby. They grow different, but actually they also, that's quite intriguing, they metabolize drugs differently, as illustrated for, again, caffeine or dexamethasone. So no worries, I still have 30 seconds, and this is my last slide, I think. I hope to convince you that be pragmatic, develop clinical trials that can be applicable to different type of drugs instead of submitting a zillion type of documents to your ERB or ethics committee, whatever it's called. Be accurate and be complete because other people would like to use your data, and the more information they have, the more robust they can be used. And believe in multidisciplinary science. It may be weird for you, but science is there to support you and actually to develop your clinical trials as good as possible and have the highest likelihood to make it into a success. Thank you for your attention.
And I'm going to welcome and transition the mic over to our panel moderator, Fabiana Dacchini.
Good morning, everyone. Thank you, Lauren, for having me here today, moderating this panel. I am one of the parent-patient partners in the IMPaCT trials, and thank you for bringing 14 of us along to this incredible journey with the IMPaCT trials. So I am a mother of a baby born at 26 weeks gestation. He's 12 years old now, and he's the reason why I'm here doing the work that I do today. And he's very complex. So a lot of things that I learned today, wow, is very applicable to us. But I want to highlight a comment that Norway made today. She said, we have to live with our decisions during pregnancy for the rest of our lives. The same apply to the decision we make in the ICU for our babies. So I just want to keep that in mind when you're talking about engaging parents in clinical trials. So we have a predetermined questions for our panelists today, and each one will have the opportunity to answer the questions, and then we're going to open to the floor to, I'm sure there's a lot of questions from you, to these amazing speakers. So we're going to kick off with Suvik and followed by Norway on the question, what makes a clinical trial meaningful?
Thanks, Fabiana, for kind of putting your life story into perspective, because I think that's the most meaningful. Is how the trial impacts the lives of the patient, and for NICU trials, the family as well. So as I had alluded to a little bit in my talk, the outcomes, I think that's the key in selecting the right outcomes. But also I tried to allude to a little bit is how it should align to the trial design, because not all outcomes that are perceived as meaningful from a family perspective may align with design. So if, for example, if you're trying to test, like it's a phase 1 trial of a new drug, then you may have to kind of look at more physiological surrogate outcomes that has an indirect effect. But if you're looking at a pragmatic trial, then yes, it should be the outcomes that you and other families have identified as the most important that will change the lives of your child. So,
Thank you.
Oh, thank you so much. Yeah, I guess to me, I might take it up like a level even higher than that and just talk about how the evidence created by the clinical trial is meaningful to people. I mean, the outcomes are important, don't get me wrong. And I would actively encourage you all to include patients, people with lived experience in selecting those outcomes in different parts or in different parts of the design and conduct of clinical trials. But I would also just try to contextualize the research question to the lives of people using that evidence, right? To, because there are some, is it meaningful to them in making treatment decisions, right? Will it address an important gap at the end of the day? And just because I think that's kind of where it becomes so practical. It takes all of these concepts and really makes it impactful to people, using that data for decision making, whether it comes to pregnancy or in supporting their child, right? With complex healthcare needs. I mean, I was once that child as well. So anyways, all that being said, I think that's kind of where the meaning comes from my perspective. So
thank you. So Isabel, for you, what is the impact of a lack of diversity in clinical trials?
So I think as we've heard about how clinical findings from clinical trials that do not include a certain patient population or participant population can then be limited in terms of their generalizability, the lack of inclusivity and diversity in research and clinical trials is going to translate directly into a lack of information that is specific and that is applicable to a wider range of individuals. So we need to think in terms of health equity at each level of the research and including in the participation. But that's where I think the co-designing of the trial and the thinking of the designing of a trial in partnership with persons with lived experience and communities and diverse communities is particularly important, because the way that information, not only about the science that it is generated by the trial, is the way that this is communicated is important, but the way that information about the trial itself and about the process of doing the trial, how to communicate that information needs to also be developed in partnership with persons with lived experience who themselves have a diversity of identities, intersectional identities, and lived expertise, because the people that are going to be eligible and are going to be included and are going to be consenting to participating into the trial is going to also be a reflection of the process and the way the trial unfolded. So when, we think about trial design, the process really needs to, that goes back to the first question. What makes a trial meaningful is not only the outcome and the impact and the evidence generated and what is going to be put into the world about what the output of the trial has been, the way that the trial has been ran, the way that the research questions have been framed, but the way that the consent material, the patient-facing material, the participant-facing material has been developed, whether it's been co-designed, whether there was input on the different baseline questionnaires, surveys that informed all the baseline demographics and all the data that will go into the trial is really crucial. And I think that's where we need to go, and think about trial design beyond the outcome and the generation of knowledge and translation of that knowledge, but start it in an integrated way.
Wonderful. Noe, can you answer the same question? I would love to hear your perspective coming from UK. The impact of lack of diversity in clinical trials.
Yeah, so I think, let me just go back quickly to the meaningful component. So one of the things from a sort of participant point of view, why it's meaningful, and just coming back to all of you here, is actually part of the reason you do it is, because you want to help others, but also from a personal point of view for the researchers. So I can't tell you the name of who delivered my baby, who gave me antenatal care, but I can give you the name of the 4 people that enrolled me and looked after me through my clinical trial. So just for you guys to have a bit of sense. So I think coming back to lack of diversity, I think it can be really limiting on what you can find out or where you can go. So again, the process is really important. Thinking about recruitment, if you don't have the right people in the first place, then you're going to be quite narrow in who it's going to be beneficial for in the long term. I think also one thing we need to remember more, especially for us that work in maternal health, is the inclusion of men. I think that's something that's often overlooked. So men also use maternity services. They come and they might just be there when you deliver the baby, but when you have a chronic condition, they might also be part of the decision-making process on what you do with your medications or even your wider family. In some cultures where I've spoken to, that it's a family decision. So, but I think in particular men are quite excluded. Where you deal with children, they're half of the product. So so I think that's really important, and just, I guess, to make sure it's relevant for everyone.
Okay, thank you. Natalie, how can we make trials more equitable?
More equitable. How can we make trials more equitable? As I discussed in my talk, and I think that this was something that one of the panelists before also said, is that think big, but incremental change is where it's at. We really have to come together as a scientific community. We have to agree to share data. We have to, prioritize areas for research and work together as a scientific community. I think if we really want to answer these questions and we want to start including, populations that have been historically excluded from clinical trials, we want to do meaningful research, we really need to collaborate with each other and kind of come, all be on the same page, and not just in Canada, not just in the US, but really, I think an international collaboration, both with regards to, how are we going to be including these marginalized populations, pregnant populations, making sure that they're diverse, and making sure that we're asking the right questions so that we really can improve the health of pregnant and lactating people. So I think it's really about collaboration.
Wonderful. Can we ask the same question for Carol? Because it would be very great to see the perspective of Europeans coming to this room on.
So thank you. I'm not native English speaking, so I asked a little bit to my neighbor some synonyms on your question. And I think I would like to simply illustrate it with one example. And if people want to read this, it's a very nice commentary or an editorial, I think in Pediatrics or in Journal of Pediatrics. It's called The Python's Embrace. And it's on the support trial. And you really have to take some time to read it. And they explain how their study got killed by additional input from the IRB. And actually, they calculated it relates to prevention of ROP. And they say, well, thanks to the way we have to communicate with our potential parents or potential patients, or the parents of the patients, the way we had to communicate with them, actually, we failed to recruit Hispanic and Black American, I hope I'm using the right wording, Black American patients. So in essence, it meant that at the end of the story, this study is actually not applicable only to white on the results. And secondly, it took more than 2 years addition to recruit all patients. So actually they calculated how many additional children became blind thanks to the ethics input. So I think this is also, I mean, even for me, an old man, white, so not qualifying for any specific aspect, it is better that we recruit or as much as possible any patients, because we will get more robust and also sooner the information we need to guide our decisions. So that's a very pragmatic, economic approach, perhaps, but it's crucial to understand we need to have everybody on board to have a relevant clinical trial.
Thank you. So let's open to the floor now. Do we have a microphone to pass around? Oh, wonderful. Okay.
Hi, I'm Joseph Ting from University of Alberta, Edmonton. Just have a questions of how, I think we all understand the importance of the perinatal clinical trials, but one of the biggest barrier or like the hurdle is about the consenting process. And especially with some ethics boards, they insist that the families need to have like 72 hours to consider for studies, and some say at least 12 hours, which I mean are completely reasonable among those like moms are under huge stress. So, but on the other hand, because of like sometimes of like we need to see the acute impact of some of the interventions, like those inotrope. So giving like 72 hours is also difficult. So is there any good kind of like thoughts about how to tackle this type of the hurdle? Yeah, thanks. From both the physicians or the healthcare and also the family perspective.
Well, thanks, Joseph. I mean, you and I both have probably spent hours talking about this and banging our heads against the wall, especially around trials on resuscitation or trials with acutely sick patients where you have a very, very narrow time window, or even prophylactic drug trials where you have to give a certain dose within 6-12 hours. And these are usually the babies with a 30-40% risk of death. Just to put the number out there compared to the risk of death in all trials that people do across all ages, where the risk of death is probably less than 1%. So, and these babies get excluded because we are essentially catering to the needs of the research ethics board's members who have been advised in a certain way to look at and evaluate trials. So I think this is probably the platform where I hope we have enough members who are like enough people who are members of research ethics boards and chairs of research ethics boards, where the discussion needs to be spurred that are we actually doing any benefit to those sick babies by excluding them from the trials, by trying to stick to the norms? And what would parents think about this whole process? Yes, we need to get consent, but for some of these questions, can we look at waiver of consent? Can we look at deferred consent? And one thought that I have is that if there are more surveys, other publications that come from a parent perspective about their willingness to explore other consenting processes, maybe we can put that on the table when we put applications to the REB to see if they'll be willing to kind of look at other consenting approaches.
Okay, thank you. I think Natalie also.
So thank you so much for that. Just to add, not only do I think we should be, revisiting the consent process, but I think we should be revisiting the way the REB structure. So, I think there were some really successful trials done during the COVID pandemic, and I think we should really learn the lessons of centralizing REB. Right now, every institution goes through their REBs, but really it could be regional or even provincial. And some, some REB centers at given institution lack expertise in pediatrics or in pregnancy and lactation. And, it's going to be quite challenging to sort of staff all of those institutional REBs with the correct expertise. But again, if there's centralized sort of hub of expertise nationally, regionally, provincially, I think then we have a chance to make some of these changes. And also, we should not be reinventing the wheel. I'm a big fan of kind of harmonizing these processes. And I hope that we would be able to do this. We'd be able to share, a consent form. It could be, as the SNAP trial showed, or, that we could, it's really a drop-down list of additional questions if you're including pregnant people, or if you're in the NICU setting, or, there should be some kind of like additional standardized, harmonized processes and tools. And this is something, again, that it sort of goes back to collaboration, but also centralizing these efforts and not kind of, all of this redundancy at the different institutional, that it ends up being really a barrier.
Thank you. Isabel or Laurie have, okay.
Yeah, I'll be honest. It is a little perplexing to me. Like, I don't understand all the different processes that happen at the REB and whatnot, but what I think what I would advocate for, beyond what you were talking about in terms of, exploring what some of these policies are and kind of designing with the community, right? Because I think there are some clear, I could talk to pregnancy, but also just even with my own children or myself over time, right? About, how ethical is it to not include them, right? It's like flipping the paradigm, because even when we talk about equitable, in the traditional sense of equality, because I know our views of equality are, evolving as they should. It's treating everyone the same, but we're not actually treating these groups the same, at all. We're actively excluding them from the process. So it doesn't even meet our definition. So I guess for that reason, I would advocate for the inclusion of the communities affected by these decisions and research ethics processes, and in designing them and in implementing them and reviewing them at the REB level. Like, I don't have necessarily a lot of experience in that space, but I know other patients and community members have played those roles. So I would just ask that if we are going to centralize, that we ensure that there's been broad engagement of those groups, that we don't lose that somehow in the process of centralization. So
yeah, so every single researcher I've ever met hates ethics. So I thought it's interesting that you brought that up. So there's just a couple of things. So I was involved in a project for postpartum, so basically heavy bleeding at childbirth and how they give the blood products. And when we did all our engagement work, it came up that deferred consent afterwards was fine. I don't know about the specifics of trial design, but one of the things they were talking about is, because there wasn't anything dodgy, it was quite like standard practices in other areas. In a couple of sites, they just changed the processes at hospital level, and they compared that with a hospital that was not using the blood products in that order. So I don't know, maybe it's also about being creative in your design. And then just a couple of other things around the barriers as well to recruitment. I think just a few things for you to keep in mind is thinking about, financial burden for patients to take part in clinical trials. I just want to make sure, we mentioned that, because if you need them to go to a site, they might have to take a day off work or time off work. You need to be able to cost in for expenses, thinking about being flexible in your design as well. If you're dealing with children, is it something that you can do, at the weekend or at school to minimize the burden for parents? And also thinking about virtually, can you use virtual formats as well? So it just made me think about some of the barriers, and for you to consider that in your design as well.
And maybe just to conclude, from the literature, we know that the decision to participate in trial as pregnant individuals or early parents is a decision that is always going to be a very personal and intimate balance of risks and benefits according to personal values and preferences that include lived expertise, culture, life experience, journey, perception of risk. And all these things are so person-centered and so individual that it becomes very difficult to think about how to make the decision process collectively. And what I would really encourage researchers to do in this area is to ask specifically, and to co-design specifically consent forms so that all the information that would be needed by individuals who are asked to take these decisions and to make these decisions in times of extreme stress, decisions that may stay in their minds and hearts for the rest of their lives, that we make sure that when we design these consent forms, the information that they would like to see is there, without necessarily changing what is needed for the trial to make sense biologically. Of course, we can always add pragmatic aspects so that feasibility can be enhanced without having a negative impact on the trial. But keeping the same parameters in place, patients, what you think participants may be worried about is probably not what they're truly worried about. So asking them at the very beginning about what they want to see on the consent process, I think, is really key to allow individual decision-making that we will never be able to anticipate as someone external to them.
Wow, that is so important. It's very relevant because I sit on the EDI for the One Child, Every Child with Dr. Bukola Salami, and she always talks about the historical abuse and lack of trust in the medical system because of history. And I think we have to understand that. Also, we talk about consent, and when I love Sylvik's picture, the young researcher coming to the room and you're so overwhelming, then I see you. To really keep the trauma-informed approach, trauma-informed care approach when you're coming to talk to families, because we experience trauma and we don't know the history that we came to that point in our lives. Many of us go through many years of infertility treatment. And so there's so much history behind that is really to sit down with the patient and understand where we are at. And then I believe sit down and talk about the consent and enroll in clinical trials. So any other questions in the room?
This is an absolutely phenomenal panel, and I really appreciate everyone's insight. So I have two questions, and the first is actually very much touching upon what you just talked about, which is there is a lot of fear of clinical trials, of interventions, of doing things on people. And how do we parlay that with deferred consent? Because I see those two as diabolically furthering that narrative, even if the goal is actually to improve enrollment. And then secondly, this is an amazing group of minds. How do we solve some of the consent issues at our REB levels? And so I've seen amazing attempts to do things like use pictorial diagrams or cartoons or other things to improve it. And that's a trial, and then you follow that with a 32-page sit-down consent to prove that somebody really actually preferred one page. And to me, that's how you actually improve access to limited communities who English may not be their first language, and particularly to some of those families in stress where they can't sit there and hear 32 pages, but maybe a handout or something to start that process would really improve it. So how do we drive that at our REB levels?
Thank you I think there's sort of two questions in there, and one of them is addressing the fear. And I don't live in Alberta, but Alberta has an excellent campaign that's sort of focused on demystifying participation in clinical trials, the Be the Cure campaign. It's really fantastic. And I think part of what we need to do is, as a community, is really, encourage and promote these types of campaigns that are directed at the general public and marginalized communities through Facebook groups or community leaders. But it's really, really important to kind of address this fear by proactively providing this perspective. And we're all sitting here in this room, and most of us are kind of in the know, but we really need to get that message out. And, you know, and that's something we can do during the consent process, but it sounds like that's not the right time. And there needs to be, you know, more of a general effort at kind of translating that knowledge in addition to obviously well done informed consent. But if the informed consent process is basically into, you know, at the way it is right now, it's like we're supposed to kind of teach them everything there is to know about, you know, clinical research and why it is important and the particulars of this study, and also, you know, paying attention to their cultural background and their perceptions and all of that, all, you know, done by, you know, one person sitting in a room in however many minutes. That's a lot, you know, so there really needs to be a multi-pronged approach. And one of it is like, especially to address the fear, I think we really need knowledge translation campaigns that really reach the general public and all the different and the diverse communities that represents. With regards to informed consent itself, I think, you know, there's lots of great ideas at this table. And I like what, you know, I like that idea of sort of picture and kind of a condensed form. So maybe like the bare minimum needs to be decided as a group, like what is that bare minimum? And then the additional kind of information in the fine print can always be added.
Thank you. Carol?
Yeah.
Sorry.
There is one potential technical solution, it's called e-consent, where you actually rather stress the process than the event, where patients can go back or can find additional information. So indeed staggered information. And I personally am happy with a no if the informed consent was accurate. So the finality of the informed consent is not a yes. The finality of an informed consent is a informed decision by, in my case, the parents in general. So that's the 2 points that I want to make. And just again to illustrate it, I was involved in a phase 1 PK study for new antibiotic, fun. 2 parents were PhD students in pharmaceutical science. So they know everything. And they said, after informed consent, Professor, very kind, we fully understand that we should say yes, but we do not dare. And so there's something that you have to respect, because knowledge and behavior are different parts of a personality. But I'm rather lucky in Belgium, patients are very interested in clinical trials. But if you look throughout Europe, there's also quite some diversity on that. So there is rather a cultural thing, and indeed informing the general public, whatever, is a way to go.
Okay, Solveig.
Yeah, just would love to kind of respond to the deferred consent issue in relation to the ICU setting, because it's a common problem that we have. And I think a lot of it is to do with the type of intervention, because in the ICU, like as I mentioned, there are probably 2 types of interventions. One is that we are testing established practices, and there's a whole lot of variation, and we do not know which one is better. Regardless of whether a child is in a trial or not, they probably have 50% probability of being exposed to one or the other because of physician differences, biases. So in those cases, when we approach it as a clinical trial, there is a fear among some people that I do not want my child to be a guinea pig for research. And sometimes it is rather difficult to explain. It depends on the research coordinator, whoever is talking to the parent, is that, well, regardless of whether you're in a trial, your child will be exposed to one or the other either way. So can we kind of look at deferred consent for some of those interventions which are accepted practices, but there is variation, versus if you're testing a novel therapy, a novel drug, and you have time to sit down and talk to families, then it is difficult. And we should not be using that opportunity to kind of push for deferred consent, where there you should be sitting down, talking to families and talk about the process. But there could be novel management approaches which are very time sensitive. again, I kind of go back to resuscitation trials. Like we follow NRP, a lot of things in NRP has absolutely no evidence base to it in the population that we apply on. And are we harming babies? Probably yes, because we do not know what we are doing, but we do not have time to explain to parents that there's this newer approach to resuscitating your child. So that is where I think, again, we need to go back to parents and kind of explore that whether in those situations, even with a novel therapy, can deferred consent be an acceptable approach, and bring it back to the RAB when we are putting our application in.
Okay.
I don't know if that would work.
Thank you. Have one more question here.
Yeah. Hello, everyone. Great presentation, very informative and very empowering for me as a caregiver. I'm a patient partner. My name is Gunjan and I live in Toronto. I have a lot of curiosity, and curiosity comes because of my lived experience as a proud immigrant and as a caregiver of a child with autism, ADHD, and asthma. I wonder, we're talking about clinical trials. Where do you see the role of psychological safety and caregiver well-being play in this journey? How are you building the foundation of caregiver well-being in this work in continuity, in collaboration with parents and caregivers? And I have another curiosity. I struggle immensely in research that we are hearing those who are always heard in the system, but we're not hearing those who have never been heard in the system. We do have a lot of EDI issues, inclusion, diversity, and equity, and many, many other issues and many barriers. I wonder, do you have any case study, any successes in reaching those who have never been heard in the system, who live in the northern and many, many remote areas of the world and have never been heard? Any case studies, any successes would be very helpful to enrich my knowledge. Thank you.
So I have an example. So for example, I work on a community-led project, research project in East London, and there's many Bengali ladies who don't speak English. And one of the things, the way we co-created this project was, I'm part of an advisory group, and we always advise the researchers on, oh yeah, this is good, but we're like, we want to create our own research. So we were brainstorming what kind of issues are there in the community alongside health professionals. And this issue of women not being able to communicate because they can't speak English was huge. So we've co-created this research project with women locally to do ESOL lessons that are targeted for health literacy. So not only are they learning English, but they're also learning about literacy and enabling them to communicate. And the step before this was that actually, even though we kind of came up with this idea, we went to the community and we had 2 focus groups with women that either were pregnant or very recently had had children and been through the care system. And it was amazing hearing this. I mean, some of them are heartbreaking, but you know, sometimes they've gone to the hospital very late because they've missed appointments or they didn't even know their appointments, so they get flagged later. You know, there was a lady who just signed something and they took her in for an emergency C-section, for a C-section, like pretty much then and there. She didn't even know what she was signing. Someone else said about, getting, like an epidural, not knowing what that was. So, we definitely realized that there was an issue and women wanted to be empowered. And one of the things as well that came up was that, initially the researcher and the clinician, they're like, oh yeah, we're going to translate all the materials. Yeah. But actually, when you speak to the community, they're like, we don't want them translated because no one is able to read, in Bengali. They want it in English because if it's in English, they can type it in and they can they can put it on their phone. So sometimes what you expect isn't quite, what might be. So maybe that's a nice example. Also coming back to the previous example as well, one thing that we found with the women was that so we're on the next stage now to implement this in hospitals. So it's been tricky. Ethics has been a nightmare. But one of the things that we're doing is using our community researchers who are bilingual, and they are consenting the women in clinics. So they're sitting outside in the, in the space where women are waiting and explaining to them, actually verbally in their own language so that it's a bit clearer. So that's one technique that we've been using.
Okay, thank you. Well, so we need to wrap it up. We have 6 minutes
Just
to
one quick comment on that.
quick comment.
Yes, I'll keep it quick. No, as an immigrant to Canada, I mean, your comments definitely resonate with me. One thing I think we should all, as researchers, we should kind of keep in mind is how we build our research teams. Because what I've found is that the research coordinators do connect at a personal level with families. And if you have diversity in there, in your research team,
Mhm.
it does help a lot, at least in my personal experience, in reaching those families.
Thank you. Okay, we can continue this conversation this afternoon. I'm sure they all be here today and tomorrow. One takeaway message from each one of you in 30 seconds each. 30 seconds.
Clinical trial conduct and workflow is culture. So it has a background on who has to develop it and also who has to undergo the clinical trial. And this is what I learned at European level. Again, there are quite some diversities, different countries on accepting deferred consent, and you can be frustrated. I feel sorry for the IRBs, but it has to do with the background and the culture, and also the legal construct is something that we as a population have made. So if we want to intervene, we have to intervene at the public level, at the highest potential level, and not at the individual level.
Thank you.
My quick takeaway from everything I've heard, and particularly in this panel, would be that the process of conducting research is as important, if not more important, than the output of the research created. So in creating that process, one of the really ways forward and the things that cannot be underemphasized is the need for partnership in doing that research. The need for, we heard, trauma-informed approaches, particularly in this area that may be talking about areas of sensitivities, of tenderness, where we may still be dealing with adversity and challenges. So I think, yeah, partnership, trauma-informed, and caring about our process and governance structure.
I think to try and make trials more equitable and meaningful, I think we've all kind of touched on it, is around involving, patients within the process. I think that's really important. And my takeaway is the importance of the multidisciplinary component. I do think that is really important as well.
I was going to basically say a very similar thing, but nonetheless, I think, to actively write in and engage patient communities throughout is absolutely critical. I think we all experience the impacts at the end of the day, but also we'll get farther together, as much as it's hard to collaborate and work together, and we all speak somewhat different language, not an actual language, but we bring different viewpoints. I think we'll get farther together at the end of the day if we all work together.
For me, I think in addition to all what has been said, spend a lot of time examining your research question, why are we doing the trial? And spend a lot of time trying to ensure that your trial question aligns with the trial design, because I think we fail a lot of times because our questions don't align with the design. We are choosing different outcomes that are not related to the question, and so on and so forth. So that is extremely critical for anyone designing a trial.
So I'm sure I will be repeating because I'm going last, but we really must all continue to work together. It is no longer acceptable to work in silos. We must work together to change culture and create a culture of inclusion. And that includes working with people with lived experience, working with communities, working with each other, and that's how we can affect change.
Oh, thank you. So thank you, everyone. That was a really wonderful discussion, and I feel in this room, we have a great recipe for success because we are all working together, collaborating, bring a different voice to the table. And I'm very grateful, Lori, that you are doing this work. And I think you're really paving the way to many other countries that want to involve patients in clinical trials. I think our voices, and I can't speak, I can speak on behalf of all the other parents, that our voices are truly heard in this work. And I'm very grateful for that. And I think we reach a point of no return. There's no way you cannot collaborate. So I'm very happy to be here today. Thank you, everyone.